I'm Chris McMaster. I'm the Director of the Institute of Genetics for the Canadian Institutes of Health Research, Canada's version of NIH. I have the very easy job of introducing somebody who needs no introduction, Dr. Eric Lander. Dr. Lander, well, he's a professor at the Broad and he founded the Broad. He's a professor at MIT and at Harvard. He was principal leader of the Human Genome Project. Played a pioneering role, of course, in understanding the human genome and manipulating the human genome. He served as co-chair of the President's Council for Advisors in Science and Technology to President Obama throughout his term. And I've got 2 pages of stuff I could read here, but I'm going to stop and introduce Eric. Dr. Lander.
Good choice. Thank you. Let's see if it's on, amazingly. Good. It's a pleasure to be here and to close out a GA4GH plenary. It's incredibly exciting to see what GA4GH has grown into. I'm very proud that the Broad was one of the founding institutions in getting the GA4GH off the ground, which clearly was a good thing, considering of what's going on with it today. It was a good idea that a bunch of us all came together, and it started with a small little group and grew to a much larger group, and you've heard this story from David Altshuler. I'm also particularly grateful to you and Bernie for taking over the responsibilities now of the GA4GH because, well, as with all good ideas, they grow, and you have to figure out what they're going to grow into and how they mature. And I really am excited about the directions that the GA4GH has been moving in scientifically and organizationally and institutionally. And so I do whatever Ewan says. And Ewan said I'm supposed to talk about the power of data sharing. He called me and said, I want you to give a talk on this. So I said, okay, I'm delighted to do that. And I am truly delighted to do that. And I want to really acknowledge, very, very seriously, Ewan's leadership in this. It's awesome. So, because it's a closing talk, I'm going to try to paint a bit of a sweep of the power of data sharing, touching on maybe some of the most important events, mostly over the last 30 or 40 years, but reaching back in one case 400 years or more, if not quite, but 350 years or so, because data sharing is a really powerful idea. So let me start with the obvious case, and the case that I know best, having this being the early part of my own career, the Human Genome Project. Now, you all know the scientific story of the Human Genome Project. I'll just briefly remind us of that. But it also was, I think, a seminal, perhaps the pivotal thing in the last century with regard to the concept of data sharing. Why a Human Genome Project? Well, because in the 1980s, methods were conceived for being able to map the genes responsible for human traits. David Botstein in 1980 in this famous paper said, hey, we could use genetic mapping to map the location of genes even though we didn't know what they did. Now, of course, Alfred Sturtevant said that in 1913, but the idea was there, but humans somehow hadn't thought about that for human genetics, and for a very good reason. In fruit flies, you could set up crosses at will of arbitrary size and mark them with genetic markers of all sorts, curly wings and, white eyes and stuff like that. And humans, didn't have wings, and you didn't have white eyes, and so you didn't have these visible markers, and so it wasn't obvious how you could ever do fly genetics in humans. And as for setting up crosses, of course, you could do it, but you had to find a nice person and settle down. And in any case, you never produced the statistically significant number of offspring. And so genetic mapping really never took off in humans until it was recognized that, forget the curly wings and the white eyes, but DNA polymorphisms would work. And the idea that DNA polymorphisms would be an incredibly powerful tool that would turn humans into a tractable genetic organism was very exciting and at the same time incredibly daunting. You could do it for monogenic traits. It was even clear that using association mapping in populations with ancestral haplotypes, you could do it for monogenic and polygenic traits. But the problem was the human genome was ridiculously big in those days. Back then, 3 billion bases was big. The genome still is 3 billion bases, but 3 billion bases now is small. But back then it was really big. And you had to find like one mutation in like 3 billion bases, and like that's a needle in a haystack, or actually we did the calculation. It's much worse than a needle in a haystack. It turns out one needle is more than 1 part in 3 billion of a haystack. So in any case, it was really hard. And that's why the world came together to do a human genome project, and went from a genetic map in 1987 to the Human Genome Project from 1990 to 2003, and it got completed and celebrated multiple times. I think we had at least 4 separate completions and celebrations for the Human Genome Project. It was a lot of work and painful at times, and so it's important to have celebrations along the way. But anyway, that's not really what I want to talk about. What I really want to talk about was that it was the pivot, the crux for ideas of data sharing. In the early 1990s, it was already happening through pioneering work of John Sulston and Bob Waterston in the worm. C. elegans, they were releasing their data before publishing the papers. And as it happens, where we were at the Whitehead Institute Center for Genome Research, the predecessor to the Broad, we're doing the same thing for the mouse. Why this was happening, I don't know, but it's sort of like we all thought, of course this made sense. And then when the Human Genome Project community came together in Bermuda, John Sulston particularly, because the late John Sulston, gets enormous credit for being a colossal pain about this point and insisting that we not only had to do it, we had to make it a principle and write it down. So John said we must actually establish principles, because we got all these groups from all the world, that we're going to release any sequence assembly more than a kilobase. That meant that raw reads were okay. They were coming off the machine. We weren't going to release them every night. But the minute we assembled them to anything, they got released. And same was true for finished sequence, immediately released. And the goal was we were going to make it available in the public domain without patents for R&D to maximize the benefits for society. So just get it out there. And John insisted on it, and we all went along with it, and it was really important. And there was an important op-ed that they wrote in Science Magazine. John and Bob Waterston did this. And there was an opposing point of view. Won't shock you to find that Craig Venter wrote a counterpoint saying that no, nobody should be forced to release their data. Anyway, that was interesting. Now, these were principles. The question of principles are, what happens when you apply them in practice? Principles only really matter when you find out in practice. So we found out not that long thereafter. In June of 2000, at the White House in the United States and at 10 Downing Street in the UK, the completion of the draft sequence of the human genome was announced. There was actually no data to support the claim that we had got a draft sequence, but it was announced that we'd had a draft sequence of the human genome. And then the question was to sit down and publish the damn thing. And as it happened, we sat down with Don Kennedy, the editor of Science Magazine, and all agreed that both papers would appear in Science Magazine back to back. That was the plan. And then it turned out that somewhere around early November, we were told, the International Human Genome Project, that Celera wasn't planning to actually release their data. That it was going to be on their website, and companies could get it by subscription, and academics could get bits and pieces of it, like 0.1% at a time you could look at. And we were pissed because we said, wait a second, there are principles here. By the way, we'd been releasing the Human Genome Project data freely, and by the way, it was appearing in the Celera assemblies. And we said, science, you're a journal. How can you say that somebody can publish a paper without releasing the data to support the claims? And I had a long number of knockdown, drag-out fights with Barbara Jasny over the phone. in which basically they tried to say, well, yes, of course, we usually require the deposited DNA sequence, but this is so important that we should make an exception. We sort of thought it was exactly the reverse. If it was so important, there should be no exception for something like this. So anyway, I was attending a meeting in New Jersey, of all places, and it so happened there was the physics editor of Nature present at the meeting, and it was on a weekend, and I just asked her, do you think Nature would be interested in the Human Genome Project paper? And she said, I'll check. So Monday morning, I'm back in my office, and I immediately have a message from Nature. We'd be very interested in said paper. And so we pulled the paper and moved it to Nature because of data sharing. We would not publish in parallel with a paper in which the data wasn't shared. Science was pissed, colossally pissed. But, tough was our attitude. You want the paper, you're going to require data release. This became such a big deal, and I say this because many people probably don't know it anymore, that the National Academy of Sciences had a committee constituted within several years to do a report whose preface says, this report is engendered by this situation with these two papers and the fact that science published something that didn't release the data. And they developed they developed a report on data sharing. And they traced this all the way back to 1665 with the formation of the Philosophical Transactions of the Royal Society, where it was, why do we have journals? We have journals because before journals, people had no way to get any credit for their claims. So mathematicians who said, I can solve a problem, the only way they could prove it is mail them a problem and they'll send you the answer. And that proves they must have an algorithm. This was highly inefficient. And so the brilliant concept of you may publish and establish, you get credit in return for revealing. And this whole notion just completely blew up the idea of credit for sharing your methods. And that's why it was such a big deal. And the Academy agreed, and they put out a set of principles that seemed like obvious to me, but they noted had not really been codified. So this particular conflict led to the codification of a lot of principles that this next generation takes for granted. So I just mention it. Now, why is it so important? And what we argued with Science Magazine was if it was the case that different people could put different restrictions on how you could have the data, we predicted that there would be a need to combine data sets. And we said, it would be chaos if there were 7 data sets that had incompatible rules about combining them. And we were right. We were absolutely right, because the genetic maps and physical maps and sequence maps were followed by SNP maps and haplotype maps and evolutionary selection maps and gene maps and conservation maps and expression maps and chromatin folding. It would be insane if we could not just lay all these on top of each other, but that was not obvious in 2001, that it was going to be the case that everything was going to be piled on top of everything, and that science would be so much poorer. But there was a moment that science got through, the world of science got through, where we could have gone into this dystopian world, or we could have gone into this world where we share data. And we shouldn't forget that we make choices about this. And a whole series of people in the 1990s led up to this moment when a choice was made, which I think has benefited us very well. So that's story number 1. Story number 2, human disease genetics. So I told you that there were these systematic principles in the 1980s about how you could map rare Mendelian diseases and common diseases with polymorphisms, tracing them in families, or looking at them for association in populations, and that was all very good. And we needed to have a Human Genome Project to do it. And then we had a Human Genome Project, and how did it go? Well, for Mendelian diseases, big success. It became a piece of cake. You have enough genetic markers, you apply them in enough families in which the trait shows clear Mendelian segregation, bingo. Thousands of traits were mapped. By today, it's over 5,000 Mendelian diseases have been mapped in this way, but these are single gene diseases. Whoops, going backward. Back. There's back. For common diseases, this was much harder. Common diseases, we now know, are highly polygenic. They don't follow the Mendelian rules of inheritance in any natural way. And so mapping in families didn't work. So this other method I mentioned of looking for ancestral haplotypes, ancient haplotypes, and we could look for enrichment of certain common variants and common segments, was an alternative. We take it for granted, but at the time, the early 2000s, people were still arguing, would this work, wouldn't it? And as with the Genome Project, we were going to have to go back to scratch and build new maps. We needed what you might call a human variation project. We had one genome, now we needed all the variation in the human population laid on top of the one genome. So we had to find all these variants, tens of millions of variants. We had to work out the whole haplotype structure. We had to work out methods for genotyping, tens of thousands, hundreds of thousands of markers, and tens of thousands of people. We needed rigorous analytical methods. And the short story is it all happened. We had things like the International Haplotype Map and the 1,000 Genomes Project and genotyping arrays, and it made it possible to do all of those things. We also, somewhat later, in the past decade or so, had new kinds of sequencers that allowed us to drop the cost of sequencing dramatically from the $3 billion we blew on the Human Genome Project to less than $600 that we have today for whole genome sequencing. That's actually a very impressive decrease in cost, if you ask me. So all that was the case, and that led to disease mapping. So using this approach, genome-wide association studies, of course, is what it's come to be called, we saw this explosion. Whereas the entire world in the year 2000 discovered one single gene associated with one single common disease, PPAR gamma and diabetes. By the way, that was David Altshuler's discovery when he was a postdoc in my lab, just mentioning since he started off this meeting. And it was tootling along at 1 or 2 or 3. As these tools got developed, it became 4, then 8, then a lot. And you would have thought people would be so happy. But geneticists are never happy. Geneticists are grumpy. And so instead of being happy, the genetic reaction was, well, yeah, but. They have very small effects. They might increase your risk by 50% or 20%. It's not like cystic fibrosis, they're not mostly in coding regions. They're in non-coding regions. That's bad. It's not good, And they don't explain much heritability, And anyway, we can't interpret them. So maybe like they're just statistical artifacts or artifacts population structure, or maybe it is really rare Mendelian mutations anyway, despite, by the way, the fact that had been failing. But intellectual consistency is a lot to ask for. But what solved this? Data sharing. It was data sharing that made this possible. Had each group tried to answer this question alone, we'd have had nothing. I'll give you a good example of it. Schizophrenia. At the Broad, we were very interested in mapping genes for schizophrenia. We rolled up our sleeves. Mark Daly and Sean Purcell and others mapped by GWAS 3,000 cases of schizophrenia, 3,000 controls, and found absolutely 0 significant loci. This didn't feel that good. And the problem is, if we didn't have rigorous statistics, we would have gotten excited about the little peaks there, but unfortunately we knew that those were not genome-wide significant. That's the problem of having rigorous analysis, is you can't declare that you found something if you haven't actually found it. The NIH study section wasn't very happy either. They said, well, this clearly fails. And 6 more groups could have done the same thing and failed also. But the international psychiatric genetics community came together and created the Psychiatric Genetics Consortium. Oh, and by the way, it's really good that some philanthropist stepped in to fund the next step of it, because the study section wouldn't have any of it. But in any case, some combination of a consortium was put together and some philanthropist. Now, why did the philanthropists believe this? Footnote: if you actually look at the 6 million p-values there and look at their distribution, if there's nothing to be found, they'll be distributed uniformly between 0 and 1, but they're not. They're bunched up closer to 0. And if you know anything, you know that means that there's a signal and it's trying to be significant, and it's saying, send more data, need bigger sample size. And bigger sample size. So with 9,000 cases and 12,000 controls, 5 loci were genome-wide significant. But the data were still saying, send more data. And the International Psychiatric Genetics Consortium pulled together people around the world sharing their cases. 5, 10 years ago, everybody hoarded their 100 cases because they thought they were going to make the amazing breakthrough with 100 cases. They said, no, together we can do what none of us can do alone. 20,000 cases and 30,000 controls, 62 loci were found. 45,000 cases, 65,000 controls, 108 loci were found. 67,000 cases, 94,000 controls, 245 loci across the genome, all genome-wide significant, replicable, pointing to all sorts of interesting biology. That is what data sharing means. We would have been, again, in a world where human genetics didn't go forward, but for the idea that we figured out how to work together and share data. And of course, today, we got more than 100,000 associations, 70,000 officially in the databases. There's another 30,000 I know about that haven't made it in there yet, for all sorts of traits and diseases. I'll mention, I was talking about the common genetic variation for schizophrenia. In the last 12 months, we're now seeing the rare genetic variation for schizophrenia also coming along here. There are now 10 loci that have been discovered by sequencing exomes that are clearly associated. Rare variants are clearly associated. By the way, 2 of them, or actually half of them, are involved in glutamate signaling. There was an old, pretty discredited hypothesis about glutamate receptors in schizophrenia having something to do with people taking the drug ecstasy, which affected NMDA receptors, getting schizophrenia-like symptoms, but everybody said, it's a shaggy dog, whatever. Looks like it's probably right. Looks like the glutamate, the NMDA receptor, and the AMPA receptors are really playing a role, and we're seeing that from the genetics. And so now we're getting a whole picture of incredibly rare, vanishingly rare things with big effect that are really interesting biologically and account for almost none of the heritability, and then almost all the heritability sitting with common variants. And there's this nice curve, I know Ewan is fond of this curve, that shows us that genetics has an entire allelic spectrum of different kinds of effects, and it's teaching us at every point of that spectrum. So it's led to lots of discoveries about mechanisms underlying inflammatory bowel disease, heart disease, schizophrenia, Alzheimer's, et cetera, et cetera, et cetera, and I could go on and on. But it's pretty amazing what's come, because every one of those cases involved international consortia of groups that have come together to manage to share their data. And not never easy, but clearly crucial. Now, I want to also mention cancer genetics, because that too has seen similar things, and I want to talk about the origin and then some recent work in this. The TCGA project and the International Cancer Genome Consortium, bringing together first folks in the US, and at the same time there's very important work at the Sanger that was going on, led by Mike Stratton, and then a whole international consortium with many different countries, and Peter Goodhand played a very important role in this, and I want to thank him for that as well. And what it did was, it said, if we look at lots of tumors and their matched normal tissues and find which genes acquired lots of mutations, we can learn stuff. We can learn about genes that are important in cancer. And sure enough, whereas over history, everybody in the 1980s was saying receptor tyrosine kinase signaling pathways were it. It turned out, no, it wasn't it. It was what you had an assay in a dish for. You could test genes for receptor tyrosine kinase signaling pathways, but you were missing lots of other stuff. And the unbiased genetic look across lots of tumors told us, no, there are genes involved in metabolism, chromatin regulators, lineage specifiers, RNA splicing. There's lots more when we could get together and begin to collect what we thought were large samples. This paper from Mike Lawrence in 2014, this is Gady Getz's group at the Broad. This was like aggregated across 5,000 tumors, which back then was a big deal. For each cancer, there were hundreds and hundreds of examples, and we could find new genes, and this was pointing to even more such genes. So this has gone on, but I want to say that cancer has also led us into new kinds of sharing. This was an important kind of sharing. Everybody going to their freezer and getting the things they had and sharing them. I want to briefly mention a new project that has arisen in the past couple of years that involves not just labs sharing with each other, but patients being partners just as much as the lab heads and scientists at the bench are partners. True patient-partnered sharing. And so several organizations have put together this nonprofit called Count Me In that enables cancer patients anywhere to share their information and samples with researchers everywhere to accelerate progress against cancer. And I'm just going to mention there are now 5 different projects in US and Canada in which cancer patients are able to go online anywhere in the country and click, enroll, sign a consent, and enroll in this study, no matter where they are in the US or Canada. There's a binational complete coverage IRB approval. And there's metastatic breast cancer and gastroesophageal cancer. There's now a brain cancer project starting. There's a prostate cancer project starting. In the US, it covers all 50 states. And what's really interesting, they go on, they fill out their stuff, et cetera. And what's amazing is that the patients have become deeply involved. Unbidden, they've started posting Count Me In selfies, where they have a picture of the spit kit. We send them a spit kit, they spit in the tube, they send it in, we can sequence their DNA. They also sign a HIPAA right of access so that the Count Me In organization can request their medical records, which are very difficult to get, but the patient doesn't have to wrestle with the hospital. They say, I appoint you as my agent. You beat them over the head. And we can also get a sample of their tumor. And so we have people who are issued very large bats that they can use to beat people over the head to get records and things like that, and they do it. But the patients themselves started posting selfies with their Count Me In box, the spit kit. I'll just read the first one. This is a woman from Houston, Texas. I want to live and watch my children grow up. But if I can't, then I want to leave a legacy and a cure. And this is really very meaningful. There's a lot of people who feel like this right now, that they want to be part of the research enterprise. Now, they're not getting their own data back, by the way, because this is not about, we're going to tell you about your tumor. They all know that at the start. This is to build. So the Metastatic Breast Cancer Project is now by far the largest group of metastatic breast cancer patients for which medical records and DNA sequences are available. 5,300 people have signed up for it. And what's very interesting is the largest institution only has 70 patients. There are 1,700 institutions represented here. And very interestingly, the TCGA project had a large number, but much smaller than this. They had hundreds, but only hundreds. But even more remarkable, it had only 3 people who had de novo metastatic breast cancer. On presentation, metastatic cancer. We've got a very large number of people showing up here, showing that a uniform sampling produced a very different picture of breast cancer. But the most amazing one I'll tell you about is angiosarcoma. Angiosarcoma is a super rare cancer. There are only 300 cases in the entire United States per year. The deputy director of the Count Me In project is an angiosarcoma survivor. She had angiosarcoma to her breast, and Corey Painter is amazing, and she knows this community, and she figured out how to reach out to this community. And we have 463 people with angiosarcoma who are registered with the project, which probably constitutes 40% of all people living in the United States with angiosarcoma. So then Corey and Nick Wagley, who's the director of this project, began sequencing, and what they found were some really interesting things. They found that people who had angiosarcoma on their head and neck had very high mutation rates. Then they said, high mutation rates are often correlated with response to immunotherapy. So then they checked the medical records, which they had gotten by beating people over the head. And they found that in the medical records, off-label, 2 patients with angiosarcoma to their head and neck that had high mutation rates had been treated off-label with immunotherapy. One in, without disclosing, I'll make it up, Alabama, and one of them in Arizona. Both showed extraordinary responses. They reported this the next month at a meeting of the angiosarcoma community, which fits in a room smaller than this. And instantly, before people left the room, they were designing a trial for immunotherapy in angiosarcoma of head and neck with high mutation rate. This information was going to remain locked in cabinets in Alabama and Arizona or wherever they were, but for efforts to share data. And that is why we care about this. This is why GA4GH matters so much, because being able to make data shareable, nobody's saying you have to share your data. Nobody in this project is forced to do anything. But it turns out we actually ask patients who are involved in this project, are you concerned about data sharing? And they say almost invariably, yes, yes, there's not enough of it. That's really the answer to are you concerned about data sharing for anybody with cancer, is why aren't we doing more of it? So finally, the last topic I will turn to is, forward, a new group that's been brought together, the International Common Disease Alliance. And this only officially began a few weeks ago in September, and I also put the Global Alliance for Genomics and Health logo here with it because this new International Common Disease Alliance views itself as very closely aligned with GA4GH, not trying to duplicate what GA4GH is doing, which is fabulous, of creating the standards for doing this, but to ask for many of the other things, for experimental projects to generate data in the laboratory, or develop new methods or other kinds of policies that aren't related to data sharing per se. We don't have a place where everybody across the community, from the folks working on genetic maps to physiological mechanisms to medicine, all come together and try to identify what's the needs list for the community. And so we met to do that. Because even though I said, oh, there's all this wonderful progress on these diseases, I'm, of course, cherry-picking the progress that we have made so far based on a limited number of the 70,000 loci, hundreds of loci, maybe 1,000 loci. But it's really hard to go from samples to variants to genes to cell types to processes to disease mechanisms. And we need a lot of work still to make this fast and easy, like it was the Human Genome Project needed to happen, and finding all the human genetic variation and the TCGA. We don't even know all the things we need, and so the community has come together to try to identify what are the roadblocks and how can we overcome those roadblocks. Sometimes it might be a big project, sometimes it might be a new little technology, sometimes it might be something else, but there's a white paper being developed to do this. Why now? Well, a whole lot of things. Human cohorts. Large human cohorts, think UK Biobank, all of this, are becoming available. Genomic technologies, sequencing costs are going to fall further. They will get to $100 a genome. By the way, if people live on average 100 years, give or take rounding, and genome costs are $100, that's like $1 a year. That easily fits in almost any major healthcare system. There are some countries in Africa whose healthcare budgets are in the tens of dollars, but for the United States, where the individual healthcare costs are $20,000 per person, $1 per year amortized is like in the noise. So we're going to see a lot of that. We, of course, have single-cell genome analysis, single-cell transcriptome analysis, CRISPR analysis, data science advances. All these things are driving us right now to saying this is the right moment for such progress. I'll just mention, no one country can possibly teach us what we need to learn. For discovering the genetics of a human disease, we're going to need large samples, and we need to look not just at disease, but disease progression, treatment response, etc. But if we want to find unusual alleles of stronger effects, populations that have been through bottlenecks, like Finland, are invaluable. If we want to find recessively acting alleles, populations with higher rates of consanguinity are invaluable. If we want to fine map populations that have shorter blocks of haplotypes are invaluable, like in Africa. And we're already seeing the biobank movement beginning to combine its efforts. Any one biobank has a limited number of people, and even more extremely, a very limited number of cases for any particular disease. But if you start combining biobanks, well, these are slides that Mark Daly and Ben Neal shared with me. What you find for asthma, when you bring together 8 separate biobanks, which have 1 million people, but between them, a limited number of cases of asthma, but much more. Any one of those countries found anywhere between 0 and, in the case of the UK Biobank, 31 loci for asthma. When you combine them all, 76 loci for asthma are found. Primary open-angle glaucoma. same sort of thing. Any one country here, nobody found more than 11 loci. That happened to be Finland. Combine them, 25 loci emerge statistically significant. And this is just the beginning, because here I quote a famous paper that projects how quickly we're going to see sequencing primarily happening in the medical community, and I believe this completely because it's Bernie et al. So Ewan says, no, it's got to be true that we're going to see more and more of this, not just in these biobanks, but in the medical system. And we're going to have to get them out with medical records and things like that. And that matters because I want 60 million cases. Sorry, that got messed up a little there. I want 60 million cases because not 60 million cases. I want 60 million participants in the combined biobanks because that will guarantee me a couple 100,000 cases. I really want a couple 100,000 cases. You saw what happened with schizophrenia, more and more cases, you get more and more loci. Well, for diseases that occur at 1% in the population, I need 100 times as many total participants in order to get that number of cases. That would be 20 million cases. Oh, and by the way, schizophrenics don't show up in your databases as often as you'd like. So we're going to need the world's combined medical information to truly squeeze out all of this human genetics, and we can do it if only we can manage to combine. And we can't just do it in Europe. We need all parts of the world represented. Also, we're finding out there's so much power in this human genetics. It can be used not just for mapping individual statistically significant loci, it can be used for building polygenic risk scores. Who's at higher risk? Which 8% of the population is at threefold higher risk for cardiovascular disease? We can use that to identify people who might get statins earlier, but we can also use that to study the genetics and, say, cell biology of people who are at high risk but not yet symptomatic and learn about processes going on. We can use it to make more effective clinical trials by picking people who have a higher risk. And we can use it to learn about cell biology by combining information from the genetics, which loci show the bigger signals, with which genes are active in different tissues, which are the key cell types, et cetera. And there are methods for doing all this. And this gets into the whole human cell atlas. Cross human genetics with the human cell atlas, and you can figure out which cell types, which processes will matter. But we have to combine tons of information. And then if we want to connect these variants to human genetic functions, observations, perturbations, biochemical rules, computer predictions, we're going to need not just the human cell atlas, but the human cell atlas times 1,000 people. And that sounded crazy 3 years ago. Now it only sounds slightly concerning. And 3 years from now, it's going to sound like ho-hum, because that's the way this stuff works. And that's going to require sharing information across many countries, many labs, many investigators. Thank God GA4GH is in the room working on these kind of things with everybody to think about how we're going to build not just one human cell atlas, but human cell atlases for many, many people in many populations. We're going to have to get in there and perturb with CRISPR and do that. We're going to have to share all the data. When you CRISPR something, how does it affect the entire transcriptome? So there's even more data we're going to have to represent and share. And use biochemical rules to predict. And then good, clean, AI-ready data sets. David Glazer here in the room is chairing a working group for the NIH on AI. And the question of how do we get data sets that are clean enough to actually do AI, machine learning on, it's not trivial. And it's going to require a lot of things, including the ability to share and clean and have common standards. And again, that's why the work of GA4GH is so important. So we need federated data ecosystems. And you guys know this, but we know this. The International Common Disease Alliance, the ICDA, is well aware of the critical importance of inverting the model. So not everybody has a copy of the data, but there's central copies in some number of clouds that people can all access with uniform APIs. And that's, again, why the work is so important, so that people can go to the data rather than the data schlepping out to everybody. And we're going to need, as you guys have talked about and we've all talked about here, federated analyses where the data doesn't leave particular jurisdictions, but nonetheless we can get answers by computing across data living in different jurisdictions. And there are all sorts of approaches now that are being developed. A number of folks are involved in this Terra data platform. But what's important about all of them is that they must be open access, open source data platforms where anybody can have access to the code. And I think we are seeing them happen. So anyway, I want to acknowledge this new International Common Disease Alliance is a lot of people from a lot of different parts of the world. It's got a 35-person organizing committee. I particularly want to acknowledge Cecilia Lindgren, who is co-chair. I'm the other co-chair of this ICDA. And we have been working to try to make this as inclusive as possible. And then particularly Rachel Liao, who is well known to the GA4GH community for the last 3 years of having been a central figure in the GA4GH community, who is over there in the room. And if you want to talk more to her, you can talk to Rachel about ICDA. So that's pretty much it. The power of data sharing. It's really powerful. We managed not to get it so wrong so far. That can't be said about other parts. Like, electronic health records can't say quite the same thing. But in this community, we haven't screwed up yet. And let's try not to screw up going forward, because, God, you just think back to the argument we had with Celera about, no, we don't want a world where we couldn't just integrate data sets. That argument was right, and we are only beginning to understand in the smallest way how right that argument is. Because we are going to see, when we have a world where sequencing is routine in the medical systems and we can exchange information, when we have federated ways to compute, we're going to see the enormous power of actually learning from the human. I am incredibly excited about it, incredibly grateful to be a member and a part and a host organization of GA4GH. Incredibly grateful to you and to all of you for being here for plenary number 7. So thank you very much. I don't know if anybody wants to ask questions or things, because it's the end of the meeting, but if there are, many want to share, but only after getting their first dibs at publishing. This sets back by a couple of years each time. What do you think can be done? Well, look, of course we understand that. And in some cases, that's perfectly fine. If you're doing one thing in your lab, publish it, that's okay. But by the way, put it on bioRxiv in advance so we get it as soon as possible. But if you're generating very large data sets of any sort, you have a public trust. The public trust is you particularly have got significant resources to generate something. That comes with a public responsibility. But it doesn't mean you have to lose credit. My first graduate student, Bill Dietrich, who built the mouse genetic map using PCR-able polymorphisms, spent half his time making a map and releasing it before his publication. And then he wrote a publication. Bill lost nothing. He was a hero twice over. He published this great paper, and by the time he published the paper, there were 100 labs already using his fruits. What we need to do to make this work is to turn people into heroes who do this. We need people to get the recognition that they get for sharing, and that should and can happen for these larger projects. An individual thing in your lab where you have a cool discovery, cool, do it. These things that bring together a whole community, we need mechanisms to recognize those people as the heroes that move the community. And we at least know from many examples it can work, and we have to simply put our minds to making sure that nobody has to choose between sharing and getting credit. How many analysts does ICDA need? Do we have enough? No, see Rachel. What are the 2 biggest gaps in data sharing? No, seriously, it's a whole amazing community. The amount of data that we're talking about, it's absurdly large. If you ask me, the gap between number of data analysts, in the field and number of data analysts needed, 2 orders of magnitude, something like that. So if you're looking for something, don't just jump right in the pool. There's lots of room. What are the 2 biggest gaps in data sharing that could be addressed by GA4GH? What part should we tackle first to realize the vision of federated data sharing? Oh, God. So I'm not going to, standing on one foot, say, here's the one thing GA4GH should be doing, because I happen to know that GA4GH is multi-talented and can walk and chew gum and spin frisbees and other things like that simultaneously. But if you ask me about one thing I'm particularly interested in, are just getting right already, it's being able to do federated analysis across jurisdictions in ways that allow different levels of sharing. I think there are extreme examples of homomorphic encryption and other things, and I think they're useful and interesting and complicated, but I'm not even sure they solve the real problem. I believe getting the right balance between super fancy math and practicality and getting instances going so people just get a comfort level. That's number one. And number two, there's only so much technology can do to protect data. We also need laws. It's just got to be the case that certain things are not allowed. I think we have to recognize that a regime of technology and regulations are appropriate. People who access certain data sets might have to actually legally say, I will not re-identify. And that's okay. And then if you're identified, you can be sued. That works. And so I think we need to get on with that question, and we are getting on with it. I know this is a question with which GA4GH is seized, but there are a whole lot of other things we need, APIs, various other things, and we are, complete in all respects from the Broad Institute to ICDA. We worship at the Church of GA4GH. What would I like to see in the future from funding sources like the NIH regarding mandating sharing from their grant recipients? Well, I would like to see them mandate sharing from their grant recipients. No, I say this not really, I don't mean to joke. A post-publication for all papers, data must be shared, period. That's what this National Academy report was about, was the data had to be shared. That's what publication means. If you publish, the data are there. You can't say, I'd like to keep working on it. Well, don't publish it yet. Once you publish it, the data are out there. That's got to be a mandated rule. And then for anybody doing anything resembling a community project, the data have to be out there as soon as possible. That means, like John Sulston said, as soon as you have 1 KB assemblies, or what the equivalent is in your world, as soon as the data are clean enough that you believe they're right and meaningful, they have to get out there. The NIH is a public trust, as is many other things, and its entire purpose turns out not to be your lab. It turns out to be the public good. And data sharing rules, when you get federal funding or government funding in other countries, should be about serving the public good. And that's okay. And again, we have to recognize people need credit, but that is not incompatible with data sharing being done early. There are ways to ensure people get credit. What would I like to, that's it. No more questions, great. That's it, was top questions 1, Slido. Okay, that was it for questions. That seems like it's it for the meeting. Good going, GA4GH. I'll turn it over to Ewan, who surely will find something to say.